into the parasitic morphology.
Wonderful. Thank you all very much for this opportunity to share some of my work with you. And so I'm coming from UCSF, where I work with Anita Sill. And so I am also a pediatrician, peds infectious disease. And one of the challenges that we faced was realizing how little we knew about some of our even basic understanding of this pathogen. And so just to reiterate some of what we talked about yesterday, cocci in the environment, it grows as, let's see, can you see you can't see that. Okay, it grows as hyphae, and then it turns into the spore form or arthrochonidia, and that can continue its cycle. And then once it gets inhaled by the host, it goes into the spheral form. And we know that this is a unique form, that it grows to this large size, has internal endospores. You can see a picture of what that spheral looks like in a pediatric patient's lung. And then it can continue the cycle. But that initial transition from the arthrochonidia choosing to go either to form a hyphae or to form a spheral is something that we can mimic in the lab with in vitro conditions, with high CO2, high temperatures, with controlled media. But that's not exactly what's happening in the lung. And so I was interested in what part of the lung or what part of the host was really able to shift this arthrochonidia to this pathogenic form. And so I've been interested in macrophages. They're key players in outcome of many fungal infections. They're rapidly recruited when you see them in the beautiful work that was displayed yesterday. And so I was interested in how macrophages were affecting cocci development. And I took advantage of a live microscope that we have in our BSL-3. And I started off with a simple question of what happens to cocci when we grow it what does cocci look like as it's growing? And so looking first at arthrochonidia that I grew in more of our tissue culture setting. So this is 37 degrees, 5% CO2 in static media. And what you can see is that these arthrochonidia swell and then they form hyphae. Alternatively, you can occasionally get a spheral that is on the smaller side, but usually you get hyphae. When I added macrophages, we saw something very different. And so here you can see an arthrochonidia in the macrophage, which looks to die like it's dying, and then these huge spherules are developing. And so this really suggested that arthrochonidia are able to sense the contact with these immune cells in order to be able to continue that pathogenic cycle. When we quantify this, we can see, when we look at the number of spherules per field, if you see with arthrochonidia alone in that first column, you see very few spherules. If we add in macrophages, you can see a drastic increase in the number of spherules per field. Not only are there more spherules, but they're also larger. So whereas in our in vitro setting and when we grow them alone in these conditions, they're actually quite small. But when we add in macrophages, they're much bigger. And we also see a delay in the time that hyphae appear in the field. So, each individual arthrochonidia gets to make a decision about turning into a spherule or turning into a hyphae, but we start seeing fewer hyphae in the field, or we don't see them appear as quickly. And so all of this really suggested that macrophages were able to, in and of themselves, promote these changes. And in work that I'm going to just summarize, we thought maybe this could be something secreted. But using a transwell assay and using supernatants, we did not see the promotion of spheral development or this delay in hyphal formation, suggesting that contact or association with a macrophage was necessary. So it could just be that the outside of a macrophage is sufficient to induce this. So contact with any part of the macrophage could have been sufficient to make the arthrochonidia undergo this transition. So I wondered if dead macrophages could really induce a spheral formation. So I killed macrophages in a couple of ways. I used heat to kill them, or I treated them with methanol to fix them so that we could expose different things on the surface. And when I did this, although live macrophages indeed could increase the number of spherules, dead macrophages, heat killed or methanol treated, and whether I added twice as many cells to increase contact, we didn't see that increase. And so and the same thing is for in terms of inhibition of hyphal formation. So again, live macrophages are able to delay that hyphal formation, but dead macrophages were not. And so this really suggested that contact or association with live macrophages is necessary to promote spheral development and delay hyphal formation. Now, one of the major roles of macrophages is to phagocytose, to consume and kill potential pathogens and to do antigen presentation, among other things. So my question was, is it really the phagocytosis by macrophages which is enhancing the spheral formation? Is it sensing that intracellular environment that is able to do this? So I treated macrophages with, so I've shown before that arthrochonidia are very well phagocytosed by macrophages. So I used an inhibitor of actin polymerization called cytochalasin D to see if by inhibiting polymerization and inhibiting phagocytosis, I could change the outcome. And so again, with my macrophages, I'm able to induce spherules. But if I treated those macrophages with cytochalasin D, you can see that we've abrogated that increase in spherules. So it really seems to be that this intracellular environment is important and is essential for them to be able to have this push to become a spherule. Now, there's a lot of different ways to understand what's happening inside or with contact with the macrophage. So we chose to look at how contact with macrophages could be changing Cocci's transcriptome to get a sense of what was happening inside of that macrophage. And so we used a dual RNA-seq approach where we're doing RNA-seq on samples from Cocci that has either just been grown alone or has been grown in the presence of macrophages at different MOIs and for different durations to get a sense of how that initial interaction is happening and how it and what we're seeing as time goes on and as we're getting more spherule development. And we chose to use a mutant that we have in the lab, which was described by Dr. Homer, RIP1, which can't make spherules. And what this allows us to do is to get a sense of how Cocci is responding and what are some responses that might be due to this important transcription factor, but also because it can't make spherules, we're able to see that spheral associated signal. And this really gives us a sign of what's happening in response to macrophages. So we have beautiful classic genes that have been shown by many members of this group that classic spheral specific genes that can be enriched in our arthrochonidia that are exposed to normal spherulation conditions, but we also wanted to see if they're up when we expose to macrophages. So I will show you here is a plot that looks at our counts per million, or the transcripts that we see in our different conditions. And so these are for SAU-GP, or spherical outer wall glycoprotein. And so what you can see here is we have in blue, we have our wild type Cocci, and we have our mutant in red. In the first columns of each of those groups, you see what the expression is for Cocci alone. And then the next three are when Cocci is in the presence of macrophages. And so what you will see is that what you can see, I hope, is that there's an increased expression when wild-type Cocci is in the presence of macrophages. And really that only happens in our wild type that can make spherules. We see that also, and even more clearly, with PSP1, OPS1, and MDR1. So that's great that we can confirm what we've seen before. Now the question is, though, are there spherule enriched transcripts that are specific to this interaction with macrophages? When we grow Cocci and the beautiful work that Dr. Homer has done, we've been able to see how spherules develop over time in our in vitro conditions. But the challenges of being with a host suggest that you will need to turn on different virulence factors or deal with different additional stresses. So what we wanted to do is to be able to find a way to compare our data sets. And so given we used different media, different shaking versus not different temperature conditions, we decided to normalize against the hyphal controls in the same experiments. So we looked at gene expression in our wild-type Cocci that's been exposed to macrophages compared to wild-type Cocci by itself. And those are the ones that were making spherules, compared to our mutant Cocci, compared in the presence of macrophages compared to mutant cocci by itself, and then compared it to Dr. Homer's work on in vitro spherules over in vitro hyphae. And this allows us to see what genes are increased in expression in these different spherules settings. And so the way that we then took all of that data is we take the genes that are upregulated, differentially upregulated, and we plot them on this upset plot. And so each of these, so the three bars, it's hard not having a pointer, the three bars that you can see from where it says 0 to 500, those are the number of genes that were specifically upregulated in each of these conditions. And then each of those then is able to be put in one of the following buckets. Either they're found in all three, which is the one furthest to furthest to your right, or they're found in two specimens, or they were unique. And we were most excited and interested about this really tight number of genes that were upregulated in vitro spherules and in our wild type spherules that were made in the presence of macrophages, because those are suggesting to be really a very specific group of spherule specific genes that may not have to do as much with elevated temperatures or specific changes in media that you see in these two scenarios, but are really conserved. And then we have in blue are the genes that are upregulated in response to macrophages in samples that can make spherules. And so we're excited about this group as ones that are able to give us a sense of what are some of the intracellular stresses that macrophages are, that cocci is having to endure while in the macrophage, and potential virulence factors. So we've looked at this in many different ways, but to give you a subset, a small version of this, we wanted to look at some of the putative secreted proteins that might be differentially expressed in response to macrophages. So there are a little over 400 genes that have signal peptide sequence that look like they could be secreted. And so we first just looked at which ones could be secreted within those two populations, and then went on to look at which might have additional increased cysteines, as was discussed previously, as a potential effector protein. And so when we look first at just the number that are secreted, we actually get a much smaller cohort with 13 in that orange group that is shared, and 21 in the group that is unique to macrophages. And of those, some of them are ones that we have seen before, SAU-GP, SOD3. And if we look at those that are small, have lots of cysteines, and are more similar to effectors in plants, those are the ones in bold. And so we're excited to dive into the role of some of these in pathogenesis. Same when we look at those that are up in bone marrow-derived macrophages exposed to spherules. And so these are really genes that we're interested in understanding what role they're playing in the pathogenesis of coccidioides, how it interacts with macrophages, and then also how it aids in virulence in the host. So to conclude, I've shown you that bone marrow macrophages induce spherule development and delay formation of hyphae, and that phagocytosis by live macrophages is truly necessary for them to be able to promote spherule development and delay hyphal formation. We're able to show that there are, and to really tighten up a group of spherule-specific genes that we see in both our samples and in our in vitro samples. And then in the presence of macrophages, we can see that cocci spherules are expressing a unique set of genes that likely represent virulence factors or responses to macrophage-induced stress. So we're further excited to look at this in a few different ways. We're interested in how macrophage factors and pathways may be playing a role in inducing the spherule development. And that could explain some of the differences. You know, when I think about a lot of my patients, there's kids that all live in the same household and only one of them gets sick. What is different about their innate immunity potentially, or other things that is pushing them in one way or the other? I'm interested in looking at evaluating candidate coccidioides genes that might be important in virulence, that may give us either new, that will be important as virulence factors. And then looking at the role of different macrophage and monocyte populations in vivo. And with that, I'd like to thank my lab, my incredible colleagues in this community, and in others, and take any questions.
So you sort of answered my question with your last slide, but I think I'll ask it anyway. Do we know which macrophages these are?
The ones that I'm using, I know, because I'm making them. So I'm using bone marrow-derived macrophages, and there's a difference in different groups that use them. And so mine are grown with MCSF. And there is that, macrophages, not all macrophages are the same. And I think that that's an important point. And I think we're learning that is even in the lungs, what is recruited. And so this is just sort of the foundation of knowing that phagocytosis can do this and figuring out how to tweak the macrophages to see which ones are then able to really eliminate this versus cause more problems is important.
So what I'm wondering if, to use the cowboy movie as a model, are some macrophages black hats and some are white hats?
I think that may be the case, and I think that that's what we're interested in. TB has beautiful literature in this, that there's some macrophages that are permissive to infection and others that are really able to clear it. And I think there's work that we've really understood in those pathogens that is beginning to have more people come in and answer some of these questions that I'm very excited about. But I think that's where we need to understand.
Wonderful work.
Thank you.
Hey, that was just beautiful. Moving away from your transcriptome for a minute, I was struck by potentially a simple experiment, or maybe not so simple. If you have to have phagocytosis to drive this spheral development, what happens, since you're going 24 to 48 hours after, if you allow phagocytosis, and then you sort out those macrophages that are Cocci positive, what happens to the remaining Cocci? Does that go hyphal? In other words, are you getting spheral development only in the intracellular to the macrophage, as opposed to, because in Candida, you will have neutrophils come and the hyphae is very easy to attack. But this is suggesting that it is really an intracellular pathogen that we haven't thought of that way before.
I think that from what I can see from my videos, it looks like a lot of the early phase is inside. So that initial development I'm seeing inside. By about 25 to 30 hours, most of those cells that are going to go on to create a large spherule do rupture and then go on to complete it. So unlike things like histo, which can grow to a large number inside the cell, it doesn't seem like you can remain in the cell forever, in the macrophage forever, and it needs to somehow escape in order to continue to create the pathogen.
Well, and that might answer, one of these questions of why your primary spherules in a host are immune masked, but your secondary and further spherules are not, because when that macrophage ruptures, now you have extracellular DNA, you have all these danger signals available to mount that immune response, and it's no longer hidden. So that's really cool.
Thank you.
Great work. We do see hyphae in humans, as you know, inside the shunt, sinuses. But I would encourage you to look into what's happening inside the cavity. Inside the cavity.
Yeah.
How do we make this fungal bulb for your future?
Yeah, no, thank you.
Because we might have macrophages there.
Yeah, thank you.
Jane, really terrific work. Just two quick questions. One, could you recapitulate what you see here with neutrophils instead of macrophages?
I need to do that.
Secondly, if we assume that in the macrophage environment, picking up a little bit where Amy left off, the environment is likely going to drop the pH in the phagosome and likely introduce ROS. Yeah. What if you did that extracellularly without including the macrophages? Could you then induce spherules formation by just dropping the pH and adding in ROS?
I've dropped the pH, and that is not sufficient in static cultures. That doesn't seem, maybe it's a combination of signals. And then the other question is, there's always nutrient deprivation and those kinds of things. I've blocked, in some of our early experiments, we've tried to block the acidification of the phagosome in a couple ways with baflomycin and with chloroquine. And both of those will result in sort of dose-dependent decreases in the number of spherules. So that is suggestive that at least it's playing a role, but whether or not that's due to its change in what the intracellular resources are versus if it's truly just simply pH is, I think, hard to know. And it may be that Cocci just needs a few signals to give it a sense if it's in the right place versus being as simple as one which we had hoped for. But